Uh, Let me share my screen right away and we'll start looking at them. I'll tell you how I handled it. You should be able to see areas of greatest interest. Can you see that?
Yes.
Yes.
Okay. Well, what I did, the questions covered a wide range of topics, and there was some of them were quite detailed. So what I did was group the various questions into categories, which you see listed here: energy, enzyme, cell division, Mendelian inheritance, photosynthesis, and oxidation reduction. I'd like to cover all of that, and if we can get through all of it today, that's fine. If we don't, we'll continue on Wednesday. And if your particular question isn't addressed explicitly, ask about it as the discussion goes on. At the end of each segment here, I'd like you to give me some feedback. Have I answered your questions? And if not, what would you still like to know? So there were a couple of questions about energy, the basics of it. What is energy? It's the capacity to do work. There are various forms of energy, potential and kinetic. Potential energy is stored. For example, in the food we eat, the energy in food is potential. Once we digest it, it becomes kinetic. Fossil fuels, water behind a dam, these are all sources of potential energy. Kinetic energy is working. For example, you convert food into energy so you can walk, or convert fuel into energy so you can drive your car, generate electricity with flowing water, heat energy for cooking. And the various forms of energy, the common ones that we encounter all the time, are heat, light, motion, electrical, chemical, and gravitational. And finally, I'd like to mention at this point renewable energy and non-renewable energy, because later in the semester we're going to talk, I hope we have time to talk about global warming and climate change as it relates to biology. On the right here are simply illustrations of chemical energy being converted into motion, radiant energy converted into chemical energy when we study photosynthesis, and so on. So are there any questions about energy beyond this brief overview? Okay, let's move on to enzymes. There were a number of questions about shape and specificity. So let's first talk about shape. What you see here is an illustration of a model called induced fit, where you see the gray is an enzyme that has a particular shape for its active site. The green is the substrate for that enzyme. Now, the substrate will fit into that active site, but not precisely. So when it does, the enzyme slightly changes shape. As you can see here, this shallow entrance is into a more rounded one after the substrate binds. So that binding then, the green changes to blue and yellow. So the enzyme reaction has occurred and the substrate has become two products, which are then released, and the enzyme goes back to assuming its original shape so that it can accept another substrate and do the reaction all over. So I have several points here with stars. Within the active site, various types of bond can form between the substrate and the enzyme. So it's not just sitting there, the substrate, it's actually bonded to the enzyme. Well, first, when the first contact is made, weak bonds form. For example, hydrogen bonds and hydrophobic attractions. After that, stronger bonds form, such as ionic bonds, covalent bonds. And here's the key thing. The differential force caused by some bonds being strong and some being weak causes torsion, which is twisting. And it's this twisting, this change of shape, which breaks or weakens bonds in the substrate. I've illustrated that with this picture below with an enzyme, an orange active site, a blue substrate. And what you see illustrated here are various bonds between the substrate and the enzyme. So this illustrates a hydrogen bond, this illustrates an ionic bond, there could be a disulfide bond. So the bonds between the substrate and the enzyme are different, and they represent different strengths. And it's this differential in strengths of bonds that causes the enzyme reaction to go forward.
Excuse me, Professor, all these bonds occurs in the same time?
Well, sequentially, essentially the same time, and it all happens in a flash, but they do happen sequentially. The weak bonds form first, followed by the stronger bonds, and it's this sequence of bonds of different strengths that causes the enzyme, or the enzyme to change shape and cause the products to form from the substrate.
Okay, and breaking the bonds and also started from the weak bond up to a strong bond in the
Let's
substrate.
distinguish between two different types of bonds here. We have bonds, if you go up to the first diagram, the green, there are bonds that hold this substrate molecule together. And some of those bonds need to be broken in order for the reaction to proceed to products. So that's one type of bond. The other type of bond is between, shown here below, between the substrate and the enzyme. So within the substrate, there are bonds holding it together. When it fits into the active site, bonds form between the substrate and the enzyme. And it's the formation of those bonds that causes the change in shape that yields products.
Thank you.
If this is confusing or not making shape, yeah,
No,
please
no,
speak up.
I got it. Thank you, teacher.
Okay. All right, anything else about that? The next question that comes up, so this is why shape is important. But shape also dictates specificity. So why is specificity important? Well, if we go down to the next statement, each cell has thousands of enzymes. One enzyme for each reaction that occurs, converting a substrate to a product. So we have potentially thousands of enzyme reactions going on at any point in time. And yet we know that many of the enzymes are off at any point in time. And so how is this controlled? If you could take an extreme case and say, what if we had an enzyme that catalyzed all reactions? So instead of one enzyme for each reaction, supposing one enzyme converted all substrates to products. Well, that would be a mess. So if you think more specifically, what if one enzyme catalyzed two pathways? And now the cell, at some point, it needs one pathway and not the other. Well, if the same enzyme catalyzed both, then you can't have one without the other. They would both be on. So that's why there's this extreme specificity between enzymes and substrates. And that specificity is based primarily on shape. Substrate has to fit into the enzyme. So do you see the point here that if you had more than one substrate per enzyme, regulation would break down?
Makes sense.
Okay, makes sense. Then we'll go on. There were a number of questions about denaturation. Enzymes can be damaged permanently or temporarily by exposing them to extremes of temperature, pH, or salt. This is denaturation. The enzyme's tertiary structure is destroyed so that it can no longer function properly. And you've seen this diagram before, where you have a normal protein twisted and folded into its tertiary shape so that it's active. And if the environment changes due to, for example, temperature, the bonds break within this tertiary structure and it unravels. So without its tertiary shape, it has no activity. Now, this is usually irreversible. In some cases, it can. If you restore the proper environment, the enzyme can go back together. That's illustrated, I think, a little more explicitly in this next picture, where you have this green thread is the functional enzyme with its tertiary shape. And the active site is represented by these three little colored balls. They're held together by the tertiary shape of the enzyme. Now the substrate has a shape that fits precisely on those little balls on the active site. Now, if you denature this tertiary structure so that it, say, is all strung out, the bonds are broken, the substrate can't fit anymore because parts of its active site are at various places that it can't reach. And I've made a little list here to remind you that what's holding this tertiary structure together are these various different types of bonds: hydrogen bonds, hydrophobic, ionic, disulfide. Some are stronger than others, but once you start to break these bonds that hold the tertiary structure together, the substrate will no longer fit. And so a denatured enzyme has no catalytic activity. And I put this graph in. There were a couple of questions about how you read a graph. So I'd like to reiterate that. On the y-axis is the rate of reaction. That's the speed with which substrate is converted to product. And on the x-axis is temperature, increasing from 10 degrees to 60 centigrade. As you follow the activity of the enzyme, as the temperature rises, the speed gets faster and faster, up to a point which is called the optimal rate. And at this point, it doesn't go any faster. Why? It's because the enzyme starts to denature. And we know that because if it wasn't denaturing, this line would extend, would continue, and the rate would keep going faster and faster. It stops going faster because the temperature is too hot for the tertiary structure of the enzyme, and so the rate slows down as the enzyme denatures. And that raises the question of activation energy, which a number of you asked about. What is activation energy? It's the minimum energy required for a reaction to occur. And catalysis is the increase in the rate due to lowering the activation energy. Now, a key point, a number of you asked, where does the energy come from for activation? Well, the source of activation energy is often the heat energy from the surrounding itself. So you can see in this graph that as the temperature goes up, the rate goes up. And that's because it's lowering the activation energy. And so for cellular reactions to occur fast enough over short times, activation energies are lowered by enzyme. Now, this graph is similar to some you've seen before, but it shows the difference in a reaction, whether or not there's a catalyst. In this case, the catalyst is an enzyme. So the purple line shows the activation energy uncatalyzed. So you can see that it takes a lot of energy, in other words, a lot of heat, for the reaction to occur without an enzyme. With an enzyme, it still takes some heat, but much less. And the rest of the force for completion of the enzyme with much less heat is the catalytic mechanism itself. That is the bonds that we've just been talking about. So the key points here are that the source of activation energy, in other words, between these dotted lines, is heat. The energy, once the activation level is achieved, the energy for enzyme catalysis is torsion, the twisting that results from the differential strengths of bonds between the enzyme and the substrate. Well, that's not intuitive. Is it clear? There are these two, the mechanism of an enzymatic reaction is based on these two factors, the heat and the bonding between the enzyme and its substrate.
Could you say that that the energy, not loss, but the decrease in activation energy from normal to catalyzed is taken up by the torsion from the enzyme? That probably doesn't make a lot of sense. Sorry.
The two together, I'm just thinking about what you said. The two
Because
together,
if you imagine the amount of energy required for the reaction to progress, if that amount is lowered by a certain amount, would that amount then be from the torsional forces?
Yes, as
Okay.
I understand your question, that you add the two together, and that's the total amount of energy that it takes to complete the reaction.
Gotcha. Okay.
Okay, anything else? All right, there were a number of questions about enzyme inhibition. Some people said they just didn't understand the differences in types of inhibition. So I'm showing you a couple of things that I've showed you before, and then a couple of new things. So types of reversible enzyme inhibitors. Make this a little bigger. There's competitive inhibition. So this illustration shows an enzyme as a blue blob. But remember, it really isn't a blob. It's really a chain of amino acids in a tertiary shape. Now, a competitive inhibitor, competitive means competing with what? It's competing with the substrate, the normal substrate for this enzyme. So the competitive inhibitor has a shape similar to the substrate, but not identical. It mimics it. It's similar enough that it can fit into this active site. And so it sits there, and while it's there, the substrate cannot go into that site because it's already occupied by this inhibitor.
Excuse me, teacher, I have a question here.
Uh-huh.
And competitive inhibitors, they maybe through medications it makes for competitive inhibitions, or by the human body itself.
Where does the inhibitor come from?
Yeah.
I'm not sure. You mean, would it come from food? It could come from anywhere. Anything that has a shape that mimics the substrate could be a competitive inhibitor.
Because it's like negative feedback that these enzymes should not be active more. Because of this, the inhibitors, competitive inhibitors, takes the part of this reaction. I don't know, it's maybe through medications, some medication for treatment of some disease, or...
Well, it could be, a medicine could be a competitive inhibitor. It could be designed by, for example, a drug company.
Yeah.
To specifically block a substrate from being converted to a product.
Yeah.
So if, for example, if conversion of substrate to product led to disease, if you could make an inhibitor that mimicked the substrate, then that could be a drug that treats the disease.
Thank you, teacher.
A similar effect can be achieved with non-competitive inhibition. So it's the same thing in the lower right here. We have this enzyme in blue, which has an active site that is the same shape as the substrate. And there is an inhibitor that does not bind to the active site. It has another site that it can bind to. So it's not competing with the substrate, but what it does is change the shape of the enzyme so that the substrate doesn't fit anymore. So competitive and non-competitive achieve the same end. It is the substrate is not acted upon, but in two different ways. One by competing, the other by changing the shape of the enzyme. And this is quite different from feedback inhibition, which is a regulatory mechanism, which when a product is needed by the cell, the pathway will make whatever is needed. But once the need is gone, the pathway stops. And why is that? In this case, we have a blue substrate and a pathway that leads into a pathway that ends up with this red box, which is the end product, that's needed for some process. And as long as it's needed, the pathway will run because the end product does not accumulate. Once there's no need for this red box, it will accumulate. And when it does, it notices that it has an active site in this first enzyme in the pathway. So if it binds to this enzyme, it blocks the enzyme such that the entire pathway is shut down. If the first step can't happen, then none of the subsequent steps can happen. So this pathway will remain off as long as this end product remains bound to enzyme 1. If there's a need for this product, then the product that's bound to the enzyme will go away and go to wherever it's needed, and this enzyme will then be active, and the pathway starts to run again. So it's entirely a function of need. The more is needed, the more the pathway runs, the less is needed, the slower it runs, or if none is needed, it will shut down completely.
Teacher, excuse me, about the feedback inhibition, it's regulate the physiology of the body, I think, by this way.
Well, it's regulated by the need for the product
In the body.
in the body, yes.
Yeah. So it's a physiologic situation. It shows. It's a normal situation.
I would say it's more of a metabolism situation. The metabolic pathways have to be regulated. You can't have them running all the time. And so feedback inhibition is one way to regulate metabolism.
Yeah, it controls things.
And I think you bring up an excellent point that some people miss is just how interrelated all of these things are. And if you don't understand the biology, you don't understand the metabolism, then you won't understand the physiology. But if you establish all of these little links in your mind, like you're doing right now, it makes everything easier to understand. So it's fantastic that you're thinking that way.
Thank you.
Okay, anything else about reversible types of inhibition? If not, the next point is about irreversible inhibition. You might wonder what could ever happen that would be irreversible enzyme inhibition. Well, it's illustrated here. An irreversible inhibitor binds to the enzyme in a way that it permanently prevents a substrate from binding. And that's illustrated in the pictures. The purple is the enzyme. The blue is an inhibitor that binds to the enzyme with covalent bonds, which are very strong and permanent. So when the substrate comes along, tries to bind in this active site, it's already blocked, and the blocking mechanism cannot be reversed. And an example of that is the way penicillin works. Penicillin is a small molecule, a beta-lactam. It's antibacterial, and the way it inhibits bacteria is that it binds to this enzyme, transpeptidase, that's required for cell wall synthesis. So if the bacterium cannot make its cell wall, it dies. And this beta-lactam binds permanently to the enzyme. That's illustrated here. This is the structure of penicillin, and this is the enzyme that the bacterium needs to make its cell wall. And what happens is, if you go over to the right, there's a covalent bond that forms between the enzyme and penicillin that's permanent. And once this bond forms, this enzyme is no longer active. Okay, so that's an illustration of irreversible enzyme inhibition. There were some questions about zymogens. Zymogens are inactive forms of enzymes. And this picture illustrates how a zymogen works. So the blue structure is an enzyme. It has an active site here, white, where this green molecule substrate wants to fit. But the active site is covered up by a substance, could be a polypeptide. So there can be, if it is a peptide, a proteolytic cleavage can remove this so that now the blue substrate can fit into the active site. And the enzyme is now active. So this illustrates how gives you an idea how to visualize what a zymogen is and how it becomes activated. So there were some questions about where in the body do you find zymogens? The pancreas, when we talked about cystic fibrosis, we know that the pancreas produces proteases as zymogens. They're activated in the intestine where they digest food. And the reason they are zymogens is if they were produced in an active form, they would digest the pancreas itself. Are there other places in the body with zymogens? And it's not clear that there are a lot. Zymogens are found in salivary glands. They're found in saliva. And also they're found in carnivorous plants. Apparently proteases in plants that eat insects are produced as zymogens. But as far as I can tell, zymogens are relatively rare in biological systems. Anybody know anything different from that? And another final point about zymogens, they are not denatured enzymes. A denatured enzyme is one that has lost its structure. Zymogens are enzymes that remain in active form, but they prevent the substrate from binding. Okay, if we're done with that, let's talk about cell division. There was a lot of interest in cell division. And several of the questions asked about similarities and differences between meiosis and mitosis. And for that, I call your attention to the worksheet. And this is a picture from the worksheet that compared mitosis and meiosis and asked what processes are unique to each and which ones are common to both. So, for example, in mitosis, there are 2 cells that result, where in meiosis there are 4. You see that illustrated here. Mitosis ends up with 2 cells. Meiosis ends up, if you go around this entire diagram, ends up with 4. There's 1 division in mitosis. There are 2 divisions in meiosis. Mitosis is somatic, whereas and produces genetically identical cells, whereas meiosis is produces gametes, produces reproductive cells. Mitosis is asexual, meiosis is sexual and is haploid. Now, what's common to both is that they both have DNA replication. This has to be, and that occurs in interphases in both processes. They both occur in eukaryotic cells. In both cases, sister chromatids separate in anaphase, and in both, chromatin condenses into visible structures. And they both have the various different phases of cell division: metaphase, telophase, prophase, interphase. So any question about that? If you want to know or want to think about commonalities and differences, you can go to this diagram. It's in your worksheet. There were a couple of questions about fertilization, and I think this figure, which we looked at before, is relevant. Meiosis yields gametes. So prior to meiosis, we are diploid. Meiosis produces female gametes, the egg, male gametes, the sperm. And it's only after fertilization that we get a diploid zygote, which then goes on to develop into a full-blown organism. There were a couple of questions about tetrads and crossing over. So I call your attention to this picture, which we've looked at before, where recombination, crossing over, only occurs in meiosis 1, in the first division of meiosis. So that would be here. And it happens when the sister chromatids, or the chromosomes, come together very tightly and they exchange parts so that you end up with recombinant chromosomes. Illustrated here, you start out with a blue one and a red one, you end up with a red and a blue, and also a red with blue tips and a blue with red tips. So this occurs only in meiosis 1, and it is a source of variation. Independent assortment occurs in both meiosis 1 and meiosis 2. So that is another source of genetic variation. There are a couple of questions about what is the role of mitosis in evolution versus what is the role of meiosis in evolution. Well, mitosis is needed for the organism to exist, in order for it to grow, to repair itself, and to reproduce precise copies, cellular copies. Meiosis, on the other hand, is critical in the generation of variation and for the production of gametes. Without meiosis, it would not be possible to generate gametes. And again, going back to look at this picture, gametes are essential for the next generation to occur. A couple of questions about the difference in cytokinesis between plants and animals. And I don't know a better way to illustrate that than with these pictures. Whereas in animal cells, which have no cell wall, a so-called contractile ring occurs between or in the middle of the dividing cell. And as it contracts, it squeezes the two cells apart so that there are two instead of one. Well, that can't happen in plant cells because of the cell wall. So the way it happens in plant cells is that there are little vesicles containing polymers, which are components of the cell wall. And these vesicles fuse to form what's known as a cell plate, which is a piece of cell wall, and then that in the end fuses to the cell wall itself in order to form two daughter cells. Now, here is something that was a problem for those of you who took the survey, the cell division survey. On the last page, page 5 of that survey, were a number of questions that almost nobody got entirely right. And the reason is that we had never covered the difference between spermatogenesis versus oogenesis. And you'd have to study the worksheet really closely to come up with the answers to this. Now, the main point that is being made here is that when sperm are made, you start out with a single cell, and you end up a single diploid cell, and you end up with 4 haploid cells, 4 sperm, that are all functional. In contrast, if you start out with a diploid cell in the ovary, and you go through meiosis 1 and meiosis 2, you end up with just 1 egg. And you don't have the other 4 products of the other 3 products of meiosis. And these are called polar bodies. So after the first meiotic division, you have what's called a secondary oocyte and a polar body, which dies. Then the secondary oocyte goes through meiosis 2, and it generates what's called an ootid and another second polar body, which also dies. So in the end, there's only 1 living cell, 1 egg. So the point is that this is a distinct difference between spermatogenesis and oogenesis. In the case of sperm, the 4 products of meiosis are all alive. In the case of eggs, only 1 product of meiosis survives. So I've copied here that page 5 so that you can see the answers in red. Meiosis produces 4 functional sperm cells in spermatogenesis. And that's the only question there is about spermatogenesis. In the case of oogenesis, meiosis begins in the embryo and is arrested in prophase 1. Cytokinesis is unequal, unequal division of the cytoplasm, producing a secondary oocyte and a polar body. You see that illustrated here in this diagram. Meiosis 2 is not complete until the secondary oocyte is fertilized. And again, in meiosis 2, cytokinesis is unequal, producing an egg, which is the only real product, and a second polar body, which dies. So at birth, the ovary of a female contains a lifetime supply of eggs, primary oocytes are eggs, that are all arrested in prophase 1 of meiosis. So that was a sort of a trick question. It was unexpected. I didn't know that this was going to be covered in this survey, but it was, and those are the answers. This is why some of you lost points on page 5. Okay, anything else about that survey? And I'd like to move on to Mendelian inheritance. There were a lot of questions about this. And Scott, could you discuss that there are really two issues? How do you do a dihybrid cross? And how do you identify gametes? So I've got those two illustrated here.
Okay, so to cut right to the end of it, the way to do these is you have to practice them. So just seeing one of these things one time probably won't cut it for most people, but there's plenty of examples online of worksheets and answer keys that you could practice with on your own. So because it's very useful to have the answer key so that you know if you're getting it right or not. But when we're talking about a dihybrid cross, right, we're looking at traits. We're looking at dihybrid means two traits. We're looking at two traits being crossed in these organisms. So you need to first, when you're looking at, was there a question?
Don't think
Okay. Okay, so when you first start with these questions, you got to figure out what traits are we looking at. And I think most people can identify that. In this case, we have a short stem yellow pea pod and a long stem green pea pod. So we're looking at two traits, one talking about stem length and one talking about color of the pea pod. And we know next we've got to figure out what kind of dominance are we dealing with. And this says long stems are dominant to short stems. Green pods are dominant to yellow pods, so we know we're talking about complete dominance. So that's also important to figure out what's going on. And when we're talking about dominance, we're really only asking what's happening to the heterozygote. And hopefully everybody knows what a heterozygote is by this point. But we're talking about an individual offspring that has one dominant and one recessive allele in one of these traits. So maybe they've got the capital L and a lowercase l when we're talking about stem length, but that's a heterozygote. So we now know that we're looking at two different traits. We know that we're looking at a complete dominance situation. So the first question is, what are the genotypes and phenotypes of the F1? This is a really standard type question because you have to do a little bit of background work before you can get to this answer. So remember, what is an F1? Well, that's the product of the offspring of two parents that have been crossed. So in order to figure out what the offspring would look like, the F1s, we must figure out first what the parents are. And then we can determine from there what kind of gametes are available, which is kind of, we'll cover it here, but it also deals with it in the second question. So we know that one parent is short-stemmed and yellow pea pods, and neither of those traits are dominant. We know we're dealing with complete dominance, so therefore we know that one parent must be homozygous recessive for both traits. And we would list that using whatever letter we choose for stem length and whatever letter we choose for pod color. In this case, we're using an L and a G. So one parent is short stem recessive, that would be two small L's. Yellow pod recessive would be two small g's, lowercase g's. So one parent then is shown as being little l, little g, little g. Does everybody understand how to get to that point so far?
Yes.
And more importantly, who doesn't understand how to get to that point so far? Because if you can't get to this point, you're going to have a real job ahead of you trying to figure out the rest of it. Okay, so no one's speaking up, so we're going to assume that people can get to this point. Now we have to look at the other parent. The other parent is long-stemmed and green pea pod. Now they tell us that it's true breeding. That's important because we know that in complete dominance, a long stem could be homozygous dominant, but it could also be a heterozygote. because just having one of the dominant alleles for stem length, for example, is enough to show the dominant phenotype. So they give us that information here with true breeding. True breeding means inbred, homozygous at all loci for the traits listed. So we know that this must be homozygous dominant for long stem and homozygous dominant for pod color, green. That means the parent, other parent must be big L, big G, big G. Are we good with how to decipher the question so far?
Yes. Yes.
Okay. So now the question comes in with, we know what the parents' genotypes are for each trait, but we have to figure out what the gametes available, what gametes can that parent produce. And this is where we use the FOIL method that I mentioned. We talk first, outers, inners, lasts, and that works very similar to the way you've seen in algebra, where we use big L as, say, one product. So consider big L to be inside one set of parentheses, and big G would be in a second set of parentheses. And that's how we get our first outers, inners, and lasts. Well, what is the only kind of gamete that a big L, big G can produce? If we FOIL that out, we will find that the only gamete combinations available, if we look at all four of them, is a big L and a big G. That's the only thing one parent can
Just one.
produce. Yes, exactly. And by the same logic, we look at the second parent, little l, little g. The only thing it can produce is what?
Little
Homozygous?
little L, little G.
Little l, little g. What was the question about homozygous?
It's homozygous because it's both little g, little l.
Correct. That parent is homozygous recessive for both traits. And so therefore, no matter how you combine the L's and the G's in the recessive parent, you can only come out with a little L and little G, which makes these kind of problems, this is fairly simple because we only cross one thing, L's and G's, big L, big G, little g. And that would show us that there's only one possibility here when we start to combine. I need to have two L's. I only have one large L available, and I only have one small L available. So therefore, my F1 generation must be big L, little l, right? Because I get one allele from the one parent, I get one allele from the other parent. So the allele for, in this case, stem length, the L's, when they combine, I will have big L, little l. Do we follow that so far?
Oh, yes.
Okay.
But I have a question about the big L, big G, big G. It's also homozygote dominant?
Absolutely correct. That parent
Yeah,
is
but
homozygous dominant.
the rest of them that are heterozygous, we should say that's recessive or dominant.
Heterozygous does not use
Oh,
recessive or dominant.
I will.
Okay,
Heterozygous means one
okay,
of each.
okay.
So that's a good question. And
Maybe
there
she
is,
means the result. The result of the homozygous will be heterozygous.
okay. Uh,
I mean, after the cross, when you get the result, it will be heterozygous.
Yeah.
100% heterozygous.
yes, anytime you do a cross between a homozygous dominant and a homozygous recessive, the answer is always 100% heterozygotes. So in this case, we see that we come out with big L, big G. We get the big L or the big G and the little g following the same logic, right? I can only I have to group my two G's together, and those are the only two available, a big G and a little g. So that must be the answer for the F1s. So I have a heterozygous at both loci, or heterozygous for both traits, big L, big G, little g. Then we have to decipher the phenotype of the F1s because the question asks us what the phenotype would be. So we know, we look at big L, little l, we know that long stems are dominant to short stems, so big L, little l must be giving me a long stem phenotype. What's big G, little g going to give me?
B, G.
Green.
Did you get that same answer, Mastura?
A long and green.
Yes, absolutely. Good job. And good job. I didn't see who popped the name up, but good job on that. Yes, it's going to be green because the heterozygote, again, it tells us green is dominant to yellow, complete dominance. Therefore, we're going to show up as a green pea pod. Now it gets a little bit more complicated because we need to know what the F2 generation is going to look like. They don't tell us it's an F2, but they tell us we're crossing an F1 with an F1. And when you do that, that produces an F2 generation. So now we kind of go back to the FOIL method to see what sort of gametes can be produced from a big L, big G, little g. Now remember, both parents are big L, big G, little g because we're taking two of the offspring from that F1 generation. So they are all identical. Their genotypes are identical. When we use the FOIL method here, we're going to see that if I combine or if I figure out the gamete combinations, I'm going to end up with 4 different possible gametes that can be produced by each parent, right? A big L, big G, a big L, little g, a little L, big G, and a little L, little g. Does everybody understand how we get those 4 gametes? And I know that the next question deals with this too. So it's obviously an issue for some people. So let's nip it in the butt now.
All of them are heterozygous.
They're not all heterozygous. When we're talking about, well, it depends on what you mean. All of the F1s that we're breeding are heterozygous, but we don't necessarily use heterozygous for the gamete. Okay, well, I'm not hearing questions, so we'll move on. Now we need to lay out our Punnett square. We know that both parents can produce 4 potential gametes. So I have 4 options for one parent, 4 options for another parent. I have to multiply those together. 4 times 4 is 16. So that means I'm going to have to do the big 16 box Punnett square. I lay out my Punnett square. As you can see on the top line, we've put in 1 possible gamete in each box. And down the left-hand side, we've put in 1 possible gamete from each box. Doesn't matter which parent is which, as long as the top line is one parent and the left side is another parent. Okay, don't mix and match, but here they are. It doesn't matter what order you put them in. We just duplicated the order that we established up front, the order we figured them out in. And from that point, we simply combine. We take our 2 big L's, for instance, in the first box, and we take our 2 big G's, and we know that that is one of the genotypes, big L, big G, big G. We figure out the phenotype. It's homozygous dominant for both traits. Therefore, it's going to be a long green, long stem green pea pod. We do that for every single empty box. We will get different combinations in a lot of the boxes as we go through. Once you have completed, can everybody understand how we're getting all of the information that goes into the boxes on these dihybrids?
Yes, we do the probability for all the options.
Okay,
And we count them.
okay, great.
Then
So
yeah.
you know that you're just combining the letters from the left side box and the top box. For instance,
Yes.
if I look at the third column with the little l big G, and say I'm crossing it with the second row, which is big L little g, I have a big L and a little l, and I have a big G and a little g, because those are the two combinations that would come up. The question asks us what the genotypes and phenotypes are, so then we have to actually go through and compile a list, and that's just grunt work where you start and go, okay, big L, big L, big G, that's the first one I've got, and I write it down and I make a little hash mark. Then I go to whatever box I feel like I would proceed along this methodically, and as long as you don't already have, you know, big L, big G, little g, for example, we don't have that in our list, so we add it in. But we do see in another place on the box, we're going to have another big L, big G, so we don't add it to the list, we simply make another tick mark saying that we've got 2 of them. Once we've gone through all 16 of our boxes, we should have a list of all the possible genotypes for the offspring, and usually we just need to total those up. I would strongly recommend you total up your hash marks. If your hash marks don't equal the number of boxes that you filled in, you just miscounted somewhere, and you need to go back and figure that out. But if we added all these up, we should find out that the numbers come up to 16, which is the number of boxes we had, and therefore that's a check to say that we have accurately totaled everything up. Make sense so far?
Yeah.
Okay, that's our genotypes. Now then they want to know about phenotypes. So then we have to go and start deciphering phenotypes. Big L, big G. We know, we've talked about it. It's going to be long stemmed. It's going to be green. So under phenotypes, we write long green and put a tick mark. Look at the next one. Look at the next one. And so add to the list if it's a new phenotype, or make a new tick mark if it's the same phenotype repeated. All right. This would be the answer that they're looking for. For genotypes, they're going to want to see a list with, it's never a bad idea to include the numbers of each of the genotypes. And then for phenotypes, they're going to want to see the actual words, right? Because a phenotype is a description. So we're going to look at the actual words, and it's useful to give them the numbers that you found. For instance, we would have had 9 long greens, 3 long yellows, 3 short greens, and 1 short yellow. Very typical when you're crossing heterozygotes like this, you'll see a 9:3 to 3:1 ratio in 16 box Punnett squares. I know it's long and it went really fast. So how do we feel about this one? Did it answer the questions that people had?
Coming up again, so make sure you understand it.
This is the kind of stuff you're going to see on your final exam, most likely. So it won't go away.
And on test 3, there were a lot of errors or omissions in trying to do the Mendelian inheritance questions. And so I wasn't surprised that when you were asking questions, many of the questions centered around Mendelian inheritance. So if you have any uncertainty, not understanding the process, make sure you bring it up or talk to Scott in the workshop or me. Make sure you understand these things. Scott, we've got only 10 minutes left in the period, so hopefully you can go through gamete identification in that time.
Okay. So let's talk about gamete identification. We've already touched base on this from the previous problem. One of the things about this question is it didn't hold your hand. It told you you want to know the homozygous parent, you want to know the heterozygous parent. If you don't know the meaning of those words, you were pretty much sunk, because it's really difficult to know what you're supposed to be looking at if you don't know what the word homozygous, for instance, was indicating. So what gametes are produced by the homozygous parent? Well, which one of these two, big F, little g, big F, big G, little g, which one of these is homozygous?
The first one, F capital, G small, G small.
None of them. I would say.
No,
So Agaret is correct. The homozygous parent is big F, little g, because although these aren't, the F and the G are different, remember we're looking at the trait, and big F was one trait, and those are the same. And little g is a second trait. Those are the same. So both of the traits we were looking for are homozygous. That doesn't mean that the entire genotype had to be all big letters or all small letters.
Okay.
Okay, so homozygous, we're referring only to a single trait at a time. So we look at big F, that's homozygous, right? Because they're the same. And little g, that's homozygous because they're the same. It didn't ask about dominant, didn't ask about recessive. It just said homozygous. So you had to know what homozygous meant. So what gametes are produced by the homozygous parent? Well, big F, little g. If you use the FOIL method, you'd find the only answer comes out big F, little g. Does that make sense?
Yes, just we should not say anything about the dominant and recessive with like this kind of homozygous.
Because the question didn't really concern itself with homozygous dominant or recessive, it was merely using the word to indicate which parent they were talking about. Because if we look at this other parent, big F, little f, that's heterozygous for that trait. Big G, little g is heterozygous for that trait. So the second parent would be the heterozygote. So it didn't matter whether it was homozygous dominant, homozygous recessive. There's only one parent here that was homozygous and only one parent here that was heterozygous. Okay. And then if we look at what gametes can the heterozygous parent produce, use our FOIL again, big F, little f in one set of parentheses, big G, little g in the other set of parentheses, firsts, outers, inners, lasts, we will find big F, big G, big F, little f, big G, and little f, little g. Again, very common heterozygote. You're going to see four basic, you're going to see four gametes that they can produce, four potential gametes. Then the next question was really trying to see if you understood how Punnett squares work. It asked how many boxes would you need for your Punnett square to account for all possible gamete combinations? Well, just like we talked about in the first problem, the first parent can only produce one gamete, big F, little g. But the second parent can produce four different gametes. So we do 1 times 4. We can only produce four genotype results. There's nothing wrong with saying 16, but it's not the best answer. And this was what you saw in the test. If you did big F, little g crossed with the four potential gametes, you got this stack of answers shown on the top off to the right that are circled in black. Now, why isn't 16 the best answer? Well, let's look at the new table.
Because we have one option, F, G, so it's the F capital and G small. And we have the second one, the second parent, they have 4. So we have to multiply with 4. As we did in the previous example, we have 4 options for the first parent and 4 options for the second parent. So we will multiply 4 by 4. But now it's only 1. So we have only one option, we multiplied by 4. So we don't have to multiply 4 by 4.
Correct. And so this second box is showing you that if you did do 4 different boxes for the first parent, the big F little g, you're going to get exactly the same answer in every single column when you did your combination. So essentially you're just wasting your time because there's no reason to get the exact same answer over and over again. Your percentages won't change. The types of genotypes won't change. None of the percentages are going to change for genotype or phenotype. So it's really just a waste of time. And on exams, when you have to lay these things out, why do 16 if you could get away with doing 4? It's a lot faster, a lot easier to tabulate, a lot easier to count. So 16 isn't a wrong answer, but it's not the best answer. The most efficient, the best answer would have been 4. 1 gamete from the first parent, 4 potential gametes from the second parent, 4 boxes.
I think, Scott, the point you made right at the beginning is even though a 16 box Punnett square is not wrong, if you came to the conclusion of 4, it indicates you really understood the process.
Yep. So now I realize we got like 1 minute left. Are there any major questions on this? And if you don't want to talk about them here, remember I have workshop coming up in 15 minutes or so. So you could always go in there too.
Okay, yeah, the time's up and we didn't finish addressing your questions. So if you go on down, you can see that the next section is on photosynthesis and oxidative phosphorylation. And so there were questions about respiration, even though we still haven't covered respiration in lecture. And so next time, we're going to pick up where we left off here, starting with photosynthesis. And we'll finish this question and answer discussion. And then the rest of the time, next period, we'll talk about respiration itself. So we're out of time, and
Yeah.
see you on Wednesday.
And excuse me, teacher, can you send us this information?
I'll put this in Canvas.
Thank you.
All right, that's the end of this presentation. I will see you on Wednesday.
See you, teacher. Have a great day.